Well, thanks for attending today. I'm going to talk about a new technology we've developed at Catalent called the GPEx Boost. It's a unique way to make high-expressing CHO cells for pharmaceutical production. The technology is based off of our GPEx technology, which we've been practicing at Catalent for close to 20 years. The technology utilizes replication competent retrovectors to insert genes into any mammalian cell line of interest. Some of the unique aspects of the technology are that we don't use any antibiotic selection as part of the process. No gene amplification using toxic compounds. The cell lines are extremely genetically stable. We've never seen a genetically unstable GPEx cell line, so that allows us to keep stability testing off the critical path for a number of programs. We've gotten titers over 7 grams per liter using the technology. And there's 13 commercially approved products that are being manufactured by our partners using GPEx cell lines. I'm going to get some data from a collaboration we had with a large pharma partner where they were comparing GPEx to their internal system for cell line development. That was a more traditional system that utilized selectable markers as well as gene amplification. The technology comparison was done using the exact same gene sequence, and we used their proprietary CHO cell line to perform the work. The only difference was how the genes got into cells, whether GPEx was utilized or their internal system for cell line development. The product I'm going to show you data from is an antibody. They could produce pretty good expressing cell lines using their system, but they had trouble producing stable high expressing cell lines with their internal system. This data just shows they took the 2 pooled cell lines. So we provided them a GPEx pool. They compared that to the traditional pooled cell line they had generated. And they isolated 360 individual clones from each of those pooled cell lines. And so the top graph shows that the titer in 96 well plates, this was static culture over the span of 7 days. You can see all the GPEx cell lines that they isolated express protein at fairly high levels. And over half of their clones they isolated from their pool did not express a product or expressed at very low levels. They attempted to use Alamar Blue to count the number of cells in each well and get an estimate of specific productivity. And you can see on the bottom graph that those trends behave similarly to the actual titer that was observed in these 360 individual clonal lines. They then selected the top 10 clones from that initial analysis, moved those forward into small-scale spin tubes where they did feeds and pH adjusted, as well as a temperature shift. So this was their base method that they used for evaluating clones at the time. You can see the best GPEx clones expressed anywhere from 3.5 to 4 grams per liter. Their best clones expressed in the 2 to 2.5 gram per liter range. So a significant difference in titer between the top clones isolated from each of the different methodologies. They then moved the top 12 clones into a relatively extensive stability analysis. They looked at the stability over 100 generations and compared their traditional clones to the GPEx clones. they considered any cell line unstable if there was a greater than 50% drop in specific productivity over those 100 generations. And as you can see on the graph on the bottom, or the table on the bottom, they looked at 12 of their traditional clones. They considered 7 of those 12 clones unstable, and they looked at 12 GPEx clones, and all of those clones were stable. So a big difference in when they compared stability between the best GPEx clones to their best internal clones for this particular product. So that gives you a little bit of background on sort of the GPEx technology and some differences we see in regards to more traditional technologies. Now I want to talk a little bit about the GPEx boost technology. And so we're keeping a lot of things the same between the GPEx boost and the GPEx technology. We're still using retrovector gene insertion, except in this time we're inserting genes into the chosen GS knockout cell line from Merck Millipore. And so we're combining GPEx with a traditional GS type knockout system. As part of this process, we're using no MSX selection. So the only selection that's being performed is the selection for GS by removing glutamine from the process. And what we saw when we combined the two technologies was somewhat unexpected. We saw a synergy between the two technologies where basically we saw a 1 + 1 = 3 kind of result in regard to titer and stability of the cell lines. So we did a comparison of GPEx boost performance to a more traditional GS selection system. And so we are using some unique IP to control the expression of GS in our cell lines and the way the genes insert into the cell line. So that's what we're calling our GPEx boost selection. And then the traditional GS selection. So we made 2 cell pools. Each of these cell pools were produced using GPEx technology. It's just the way the selection was performed that differed between the 2 groups, the traditional GS selection versus the GPEx boost selection. You can see the 2 pools we generated were about the same gene copy number, but we saw a dramatic difference in final titer, where we saw about a 2.5-fold increase in the final titer of the GPEx boost selected pool, as well as a 2.5-fold increase in the specific productivity per copy of the gene inserted between the GPEx boost selected pool and the traditional GS selected pool. So with approximately the same copy number, the GPEx boost technology led to a significantly higher expression and specific productivity per copy of the gene inserted. So I'm going to show you data from 3 different experiments we ran where we expressed 3 different proteins. One was a difficult to express FC fusion protein that we had a little bit of trouble getting a really high expression from in our GPEx cell lines. One was a more difficult to express IgG1 antibody, and then we chose a medium to high expressing IgG1 antibody product. We produced all these products originally using our GPEx technology. And just to highlight again, the only difference between the experiments was that we're using the chosen cell line compared to our base, which is a CHO-S derivative cell line. And all the regulatory sequences, et cetera, all the gene constructs were kept consistent between the two technologies. The only difference was the addition of the GS gene and how we were performing selection for the GPEx boost technology. This slide shows the data for the FC Fusion product. Again, we had shown this was relatively low expressing product when we used GPEx expression. And so the table here in the graph compares the expression level for the GPEx boost pool compared to the GPEx pool. So you can see the GPEx boost pool performed substantially better from a titer point of view, with titers of a little over 4 grams per liter compared to about 0.6 grams per liter for the GPEx pool. When we looked at copy number in this pool, we saw a slightly higher copy number in the GPEx boost pool. Specific productivities were significantly higher for the GPEx boost pool, about close to 3-fold higher. And specific productivity, again, per copy of the gene inserted was higher for the GPEx boost pool. So not only are we getting higher copy number in the pool, higher titer, higher specific productivity, but each of those copies inserted is performing at a much higher rate, where we're seeing higher specific productivity per copy of the gene inserted. One thing to point out is when we did optimization, clonal selection and optimization for this particular product using the GPEx technology, the best expression we got was about 1.8 grams per liter, and the specific productivity of 11 picograms per cell per day. So we saw significantly higher expression already in the pool, even before clonal selection, for the GPEx boost technology. For the next product, this was the relatively low expressing antibody comparison that we performed. Again, here we're looking at a measure we use called total gene copy index. So you can see the total gene copy number is relatively consistent between the two pools, but we saw a significant increase in the expression of in the GPEx boost pool, where we got about 1.8 grams per liter compared to about, again, 0.6 grams per liter expression. And when you look at specific productivity, again, we saw about a 2-fold increase between the two pools. So GPEx boost performed at about 2-fold higher level of expression. Again, when we did clonal selection on the original GPEx cell pool and optimized the clones, we got the clone expression up to about 1.8 grams per liter. So basically the same level of expression that this new GPEx boost pool is expressing at, even before clonal selection. So then the next data set, we looked at the medium to high expressing antibody. The GPEx boost pool expressed at about a little over 3.5 grams per liter. The regular GPEx pool was about 2 grams per liter. In this instance, we didn't see the big change in specific productivity between the groups, relatively similar specific productivities, just a pretty dramatic increase in titer for the GPEx boost pool. So in summary of this data, for the pooled cell line production, for the FC fusion, we saw a dramatic increase in expression of about 7-fold increase. For the difficult to express antibody, we saw about a 3-fold increase in expression when we compared the pooled cell lines. And for the relatively high expressing antibody, we saw about a 1.8-fold increase in expression when we compared the two pools. So what do we think is occurring in the pools that we're looking at? And so this slide just is a illustrative example of sort of a more traditional methodology, which I showed you earlier that we compared to our traditional GPEx technology, and then what's occurring with our GPEx boost technology. So for standard cell line development technologies, when you're originally isolating clones, you tend to see a lot of clones that are low expressing, and you have to look very hard to find those high expressing clones. So this was I illustrated earlier in the comparison between the two technologies, where a lot of the clones didn't express or expressed at low levels when our partner was using their internal cell line development technology, but they were able to find some clones expressing at pretty good levels. With the traditional GPEx technology, as I showed you in that example, all the clones expressed, and they express around a median, which is the pooled expression level. So you see all the clones expressed, and they all tend to express around a median. So you tend to see more of a bell-shaped curve in the expression level of individual clones. But with the GPEx boost technology, we're shifting that bell-shaped curve significantly to the right and doing sort of an added really hard selection for those cell lines that are really producing high levels of the product of interest. So are the GPEx boost cell lines stable? This was a study we did. We had another reason for doing this. We wanted to see if we could make pooled cell lines that were stable in their expression pattern. And so we took the two antibody products we made GPEx boost pools for, and we took those pools and put them under a stability study. So we did about a 40 generation stability study where we evaluated titer throughout at different time points throughout that approximately 40 generation stability study. And you can see even the GPEx boost pools in this example are extremely stable. So we saw virtually no change in the production level for either antibody over those 40 generations. So this was a somewhat surprising result. We were excited about this because we have some potential applications that we're looking to use pooled cell lines for, and having stability will allow us to do that, use them for some of those different programs that we're looking to move forward in development. So our whole goal in developing new technologies at Catalent is how can we shorten and make more efficient the biologics development timeline. And so the process we're using now for our cell line development, as well as our process development for products that want to go fast, and in this the current age we're living in, with COVID products being with a number of folks wanting to move COVID products quickly into the clinic, we're using this process for a number of those products to speed the development cycle and to move them forward as quickly as possible to hopefully get them helping patients in the near future. So like any program, we start with DNA cloning, and then we do our GPEx boost cell line development. We end up with a pool of antibody producing cell lines if the product we're making is an antibody. I showed you those pools can be scaled and are stable, so we can make large quantities of material from those pools. And at Catalent, we use the Berkeley Lights, Catalent, we use the Berkeley Lights beacon technology to use microfluidics to quickly identify and isolate high-expressing clonal cell lines. Typically, we're looking at about 1,500 clones at this stage. We select the best clones to move forward, and once we expand those clones, we select usually the top 12 clones where we take those directly into amber bioreactors. So these are 15, an amber 15, so 15 ml mini bioreactors where we can, if we take 12 clones into the amber, we can look at 4 different conditions. So get a quick read on how those clones are responding to different medias and feeds. And then once we identify the best clone and the best condition, we can scale those processes up directly into single-use bioreactors, which is our base platform at Catalent. So when you want to go fast through the development timeline, you want a cell line technology that has a number of features. One, you want it to have high yield because most of these products, especially if they're going to be COVID related, are going to be needed in very large quantities. And so high yield is going to be beneficial for those particular products. You want shortened development timelines, so you want a cell line technology that goes fast. And then you want cell line genetic stability. So you want that cell line to be genetically stable so you can keep stability study off the critical path and know that your cell line is going to be stable. And again, the middle panel just highlights the GPEx boost technology and sort of this unique selection we're seeing where we're getting very high expressing stable pool cell lines, which makes it easier to identify those high expressing clones. And so the panel on the right is the FC fusion example. I showed you the data from a little bit earlier, where we saw stable pooled cell line expression around 4 grams per liter. We took it through that fast development process where we took the best clones, put them into the amber bioreactor. The best clone and condition that came out of that amber bioreactor was a little over 5 grams per liter in the amber. And then we took that directly into a 50-liter single-use bioreactor system in those same conditions. And you can see in the bioreactor, the process scaled very well. We saw a little bit of an extended culture once we got in the bioreactor and things were controlled a little bit better. And we actually got a little over 7 grams per liter titer for this particular product. And so this is just an example of when you take something fast through the process using the GPEx boost technology, you can see these very high titers at the pooled stage, at the clonal stage, and it scales directly into the into single-use bioreactors at larger scale. So with that, I'd like to conclude. The GPEx boost technology results in significantly higher titers than traditional GPEx technology. Difficult to express proteins perform particularly well in this system with up to 4x higher titers, and in one example even higher. Even the pooled cell lines are extremely stable with the GPEx boost technology. So we've seen this exact same stability profile as we do with the traditional GPEx technology. And these improved titers are a result of higher gene copy number, higher specific productivity per copy of the gene inserted. And we're also seeing an increase in the integral viable cell concentration of this particular cell line in the conditions we've optimized for the cell line, where we're seeing slightly higher cell numbers that are leading to those higher titers. So with that, I'd like to thank you for attending and would be happy to answer any questions that you might have.